Hello there, and welcome to Preprints in Motion, the podcast taking a deep dive in the fast-paced world of preprints.
Join us as we sit down with early career researchers, discuss their latest preprint, and find out about their journey through the muddy marshes of academia.
Hit that subscribe button, leave a rating, and support us by heading over to buymeacoffee.com/preprints. But for now, let's get into the show.
And today we're talking all about sperm, from flies to humans, with Rion and Joana.
So, we're going to be talking about a preprint that's gone on to be published in eLife, which might be the first one we've done actually with their new model. So we'll probably talk a little bit about the new model, but we start in the same place with everyone, which is if you could just introduce yourselves, a little bit about your background, and then we'll move from there.
Okay. Can I go or do you want to go, Rion?
Go ahead.
So I'm Joana. I'm a PhD student. I'm currently in Lisbon, Portugal, and a little bit of my background academically. So I did my bachelor in molecular biology, and then I wanted to get out of the academia scene. And then I changed to another university, Technical Institute in Portugal, in Lisbon as well, but to do biotechnology. And then I missed the bench a lot, and I came for a PhD.
All right. My name is Rion. I am originally from Brazil. I did undergrad and master's in Brazil, information systems, then business. Then I fell in love with teaching. I got an opportunity to teach while doing my master's, fell in love with teaching the students, the spark that brings in their eyes when they learn something. Decided to go for a PhD, came to the US to do a PhD in complex networks and systems at Indiana University. Was there for 6 years, and then once, because I wanted to continue in academia, I went to a postdoc position. That's where I met Joana, and we can talk about that whole story. Stayed there for 4 years. Didn't go into academia as I would like to. Ended up shifting my career into industry, or back to industry. I'm now work for a really cool company that's called NielsenIQ. It's a global company based in the US, but I work from the Portugal office in Lisbon.
I love Lisbon. I miss Lisbon. Maybe that could be my holiday again this year. I was there in October last year. It was lovely. Okay, so could you jump a little bit into what the preprint paper is that we're talking about? Just a broad overview, whoever would like to take that big task.
Okay, so it's a preprint with a lot of collaborations. There's a lot of people involved. I can specifically talk about my part and then Rion can complement. But basically what we did was to look at let's call it the cell identity of a sperm cell. So we started by looking at an evolutionary view to see what was composing the sperm cell per se, the masculine sperm cell. And what I did was then to look at a specific time point across the development of the sperm cell. It was the transition from mitosis to meiosis, because that was our specific interest. And then I analyzed a bunch of genes, and I was amazed by how many were contributing to that specific stage. And then we crossed that data with the human data, and Rion did a bunch of work on bioinformatics behind.
So maybe if I can add a little bit to that. Coming from a complex systems background, we are interested in like networks and how individual genes have to work in common or in conjunction, so that as a whole, it's much bigger than the single genes individually, right? So we're interested in like, how does this blueprint of genes in the network all the way interact among themselves? And there's like this complex thing, this black box that happens. And then on a phenotype level, you see a completely different organism, or you see, in the case of the problem, a motile or like a sperm cell that doesn't move, right? How do you go from that gene level all the way to how the sperm moves itself? That's just one of the cases. Joana, you can go into the details of the different classifications of like healthy and unhealthy sperm cells. There's a full range of them, but I'm interested in how we go from like individual gene into the network and then to the outcome, right? And my work has mostly been domain agnostic. So it doesn't only apply to biology. You could talk about, for example, individuals in a company, or you can talk about individuals in a country. They're all individual things, then work together in a network to come up with a phenotype. And that can be all the way from biology to social technical systems.
Yeah, that was one of the two things I really liked about this bit of work was that maybe this is my background as an immunologist, and we only like to do one thing at a time, unless it's CD numbers, and then we like to do all of the CD numbers because there's our paper title for us. Um, but we generally will knock out a gene, and then you've got a paper about a single gene, and people kind of forget that it's not just these things working in isolation. There's a lot more going on, and I like the network approach. The other thing that you did that was really cool is you went from flies through to humans, which is not something you see people doing very often either. And again, I think it's a really nice way of we keep talking about doing proper, rigorous, good quality science. And I think that's kind of what that is. That's what it means to me anyway, sort of going through the full process. Because you could have just stopped at flies and not carried on if you'd really wanted to. How do you find doing that kind of work across multiple different samples? Is this because it is so collaborative that you can do that, or do you have to handle lots of different types of sperm? Is sperm majorly different? I'm asking lots of questions there.
Well, actually, sperm from flies and human are quite different. The sperm cells from the flies are very long, and instead of the bulb-like head that we see in humans, they have a needle. So it's very different to distinguish. It's very hard to distinguish from the head from the tail just using optical microscopy. And yes, it was a ton of work at the time. So we were very pleased to have a collaboration with Germany, Münster, the institute that Frank Tuttelman works at. And what they do have is the exome sequencing of infertile patients. So we knew that we were interested in patients that had a phenotype, so they could not reproduce. We didn't know a priori which kind of problem they were having, because it can be either a genetic problem or an environmental one, or it can even be something from the family background. So what we did was to focus on this specific burst of genes that you see in the transition from mitosis to meiosis. And then when we had that data, we started with around 900 genes, and we silenced each one specifically at meiosis entry. So we used RNA interference to do that. And after having our 250 flies that had a reproductive impairment, we crossed that data with the human data. And we were trying to see if we could filter some of the variants out, and if we could focus on finding a new genetic cause for male infertility. Well, the flies are very helpful, but they are a first step to approach this big problem that is infertility, that we see more often nowadays. People have kids later. Men don't have as many precautions, let's say, as women do, because we were conditioned to think that the problem was always from the woman. So we are very careful in what we do throughout our 30s or our late 20s, and men are more relaxed in that aspect. And we are trying to change that mindset as well, because we now know that the age and the lifestyle also has an impact on sperm cells.
It's all that 5G we carry around in our pockets on our phones. That's what the conspiracy theories would tell you. It's not 5G, obviously.
No, but Tupperwares are a very big thing. And also the paper that you get from the stores, the receipts, these are also a big thing. So be careful with the receipts.
Yeah, that is actually true. I don't think many people actually know about that. Could you just describe what the difference is in the phenotypes you're looking at? So what makes a sperm healthy versus unhealthy?
So here we have some different approaches that we took. So the first step was to silence the gene that we were interested in. And then we crossed those males that have the silenced gene with females that are well-typed, completely normal, completely fertile. And we attribute them a rate for the fertility. So we have our own approach based on our control situations. If it's below 75%, we consider the gene important for fertility, and then we dissect the gonads of the male, and then we, on a microscope, try to understand what's the basis of the reproductive impairment. And here we have several categories. If you're not keeping up, please let me know. So we do have a pre-meiotic category. So cells stop developing before entering meiosis. So what you usually see here are what we call vestigial testes. So they are very small. They are only comprised of somatic cells and early, very, very early germ cells. And then the germ cells can commit to meiosis, and meiosis can be impaired. And here you have a meiotic arrest or a meiotic death, because we cannot, without a proper staining, distinguish between the two of them. So here you have the pre-meiotic cells and the meiotic cells, and then the development is stopped. And then you have what we call an individualization defect, because in Drosophila, the sperm cells develop in clusters of 64 spermatids. So when you already have the head of the sperm and then the tail is forming, it's what we call spermatids. This comprises a lot of morphologicals and cytological differences. So if we see that, so the whole test is seem very normal, and then in the seminal vesicle where the flies accumulate the sperm to inject into the female are not there, so the seminal vesicle is empty. We call that an individualization defect. And then lastly, we have a gamete defect, and we can approach this by just pinching the seminal vesicle where the sperm is stored, and by eye try to analyze if there's a slower motility, or the sperm doesn't move at all, or if it's a lower amount of sperm. And then, of course, we have scientific, very rigorous tests for all of this. Because we had 250 genes on hands, we didn't do for every one of them. So there's a lot of stuff that people can get from our paper to still try to squeeze out all the genes that we approached at that time. So please, for the people that are going to hear the podcast, please do it, because I would love to know what's going on in there, and I don't have the time.
I mean, that hands it nicely over to the coding side of it and the data part. It is a really nice match between the sort of wet lab stuff and the computational stuff. How did you both find that communication? Because sometimes you get labs where that communication isn't the best, and so the wet lab people can't really quite communicate with the computer people, and then the other way around. Other times, obviously, it works great. How do you find translating things across?
Maybe Joana should also say something on this, right? But at least from my perspective, the thing I love the most is that we were very close to each other, right? So I was just a few doors away, and I will come with the lab with Joana and Paulo, and I will get in and say, "All right, what are you doing today?" Right? Can you show me exactly what this means? It's like you're saying it doesn't move. So what does it look like? And then I would put my eye on a microscope. It's like, oh, okay, so these are moving, these are not moving, right? So how do you get the 75%, right? How do you measure? And then they explain, oh, we do multiple times. And then you understand the complexity and then like the scale of how many flies you need, how many tests you need on the same type of flies with the same type of genetic alteration. So how do you go from like the individual fly analysis to the robust statistics, and then to the complete set of all the 900 genes that we had to look at, right? So you understand the process. I think that's to make it easy, you need to understand the process from the beginning all the way to the end. And I think that was the great collaboration that we had, at least from my perspective, to understanding the biology that I have no background on it.
And also for me, it was very nice because, as Rion said, he was just a few doors away. So anytime we would meet, I don't understand anything about, well, computers at all. And he was trying to explain everything very well. To put it in simple words for me to understand, because I don't have any background on bioinformatics at all, and make it do a little jump between, okay, but we cannot do it like that because the way that I'm doing is A, and then we are thinking about B. But so if we can get middle ground for both of us, it was perfect.
Yeah, I'm jealous. I had to teach myself bioinformatics in my last project, and I'm not sure it went well. It still hasn't been published, so I can probably say that for now. But while we're on bioinformatics, could you talk a little bit about the gene networks and the ortho backbone, which is a word I did not know, and sort of what role that played in the project, a bit more?
That's a very good question. So the lab I worked on and pretty much my research throughout academia is mostly being involved in networks, right? These can go, so here we're talking about the genetic network, but this could be an airline network. This could be a network of friends or social networks. It could be offline networks. It could be online networks like social media, Facebook, Instagram, all of that. So we are interested in not the things themselves, but the relationship between the things, right? And that's why networks are so important in biological systems, because the network of how things interact is often as important as the things themselves, in our case, the genes, right? The lab I worked on has worked in a lot of methods to try to distill what is important in a network, right? Just like you would do instantly understand what is the most important relationship you have with your friends. So this is the friend I like the most. This is the friend that we talk a lot. This is the friend I go for bioinformatics questions, right? How important your network plays a role in your life. We wanted to understand how the network of genes played a role in detecting which genes are important for the meiotic defects, right? So that's what we wanted to study. And we applied, so first we built the network, right? So we had huge networks. We were started with all the genes, and then we distill down to the important genes detected that were on a threshold of being expressed or not expressed. This network is very dense. So how do you go about analyzing? It's pretty much every gene is connected to every gene to some degree. So there is a methodology we use called the backbone that distills what are the most important connections you need in order to have a shortest path approach. So if you need to go from you to someone's best friend in, let's say, in London, now what's the shortest path you can take to get there? Or if it's a street network, how do you go from your small town all the way to London on the shortest path, right? So that sort of thing. That's one of the methodologies. We tried a few. So you could go by the shortest path. You could go by not taking the highways. You go by train. So there's different networks you could use to define what your shortest path or most important path is. And that's exactly what we did. So we removed away a lot of the noise and we stayed with the most important connections that for us made sense, right? And that's exactly that drove the science in also the interactions between us to say the network is telling us that these other genes that are closely related to the gene we see has the problem or is the one responsible for the problem, maybe we should also investigate these other two genes, right? So it's not about the individual gene anymore. It's about the network and the relationship of that gene we know about the neighbors or about the ones that are in the same path.
And as someone who loves Drosophila, because that's what my PhD was on, talk a little bit about why you use Drosophila for some of this work.
Okay, so that's an easy question. Come on, you're a Drosophila guy, so it's cheap. Let's take the obvious, because doing this work in mice, it would be very, very long work, and then it would have to maintain the mice. In human, it's not ethical, so we can't go for that approach. Any other mammal would also be very, very difficult and expensive. So Drosophila, although I now find it a bit expensive to buy the flies, but we do have a lot of margin to work with high volumes. Well, the life cycle then, it's quite fast, so it's very suitable to the PhD time, and it's very, well, it's conserved with humans. So we see that it's not a one-to-one ratio. So you don't have a gene in human and then you have a gene, an orthologous in the fly. Sometimes it's 3-to-1 or 4-to-1. It depends a lot. So we have to be very careful when looking at Drosophila because of that. But besides these small constraints, yes, it's basically very fast to grow, quite cheap. We can maintain it very easily, and then we can adapt the genetic alterations that we need to make. Do we need to grow them in an incubator so that they express this gene, or do we need to put them in a colder place so they express that gene? So we do have a lot of margin to work with that.
I have to ask. I like people talking about how great Drosophila are. Everyone should use Drosophila. Although I do not miss coming home with flies in me and on me. It's a risk you take. I had one Christmas where we were tipping flies and I had to take them on holiday with me, which is a weird thing to do, to just carry files of flies around with you.
Yeah, on the pocket, on the train. Sometimes I find them in a hair. They had one in the
Yeah.
hair.
Yeah. They do come home with you.
Coming from outside, it was super cool because I would get in the lab and they were all with like this huge vials of flies. I was like, what are you doing today? Oh, these are the vials for the gene X and Y. I found that like amazing, right? So for me, it was like a great experience to walk into the fly lab and to see them with flies all over it.
All the non-scientists, like when I tell them I can sex flies and tell them if virgins or not. That's a really useless party trick I have. It's never yet come in use, but maybe one day. You never know.
Yeah, I think that's a joke that everyone
Yeah.
makes with Drosophilists, is that while they're selecting your virgins. So, although I have some troubles in the recent times that they are still very young and they're already accepting males. So I'm like, come on, girls, this is not the way we do it.
You need to play different lab music to them, clearly. Um, put some really boring audiobook on. So how do you see this kind of big data knowledge that you've generated translating into a clinical kind of setting?
Well, that's a very hard question to answer because I cannot say A or B. So, what we want, the major goal of Drosophilus in male fertility is to uncover new genetic causes of infertility. So, ultimately, the goal would be for doctors to have the already very well established guidelines. So, when they're looking for some kind of alteration in the DNA, they can also look at that gene that we uncover that we know that does A, B, and C, and when disturbed, it causes X phenotype. So that, of course, would be the end goal. It's a very laborious process. It's not going to take 4 years or 8 or 12. We have to approach it step by step, gene by gene, and then get into the clinics because sometimes older doctors are very reluctant to accept that this new gene has an impact because they don't have the clinical experience that tells them so. So they go for the first line guidelines that were approved, they stick to them, and then we have to do some forcing to push them to a fertility clinic or an institute in a university where the sperm can be studied. Because it can be very simple to treat if we know the baseline of the problem.
And you were talking about wanting people to take a look and sort of build on what you've done. So how important was it to have you to have that data publicly available? And how difficult was it to wrangle all that? Because I know my code is an absolute mess because I'm a lab biologist, but my code is horrible. I hate sharing it with the world. But how did you find that process of making it actually usable?
So that's still a little bit out of my hands. We published the paper and we are hoping the clinical setting is taking a look at it. We know that our collaborations are aware of the genes that might impact fertility. So, for instance, when we look at database of genome or exome sequencing, we have a lot of mutations that we can look at. Some are relevant, some are not, some are related to the fertility, others are not. So we do have to have that filtering. But if I'm a doctor and I'm presented with that database, and I know that this gene has an impact on fertility, this is going to be one of the first variants I'm going to look at. So that's a very important first step that sometimes people overlook. But well, I think that I hope actually that my work contributed to looking at those genes with a little bit more attention.
Also, the platform we built, right? So we built a platform to make it easy for people. It means like other researchers or medical doctors, anyone really that wants to explore the additional results that we have, like the full list of genes we worked on. They can easily navigate and access. It's a website we built, and they can navigate the results, see what are the levels were for fertility for that one specific gene, right? And they can open the networks. If it's a bioinformatician, someone that knows how to reproduce code, they can go into the code that we made freely available on GitHub, including the results, and they can reproduce the same tables, the same results, the same plots. Everything we did, we made it open source to the community so that they can build upon.
What are your thoughts on sort of having that website angle to it and how important that is? Because this is something that is increasingly happening, particularly with large data sets. And it's one of those skills that a lot of labs don't necessarily have. Like I said, right, I was teaching myself how to do bioinformatics. I wasn't going to go make a website for it as well. So how important do you think that is as a science communication tool and for that reusability angle?
Um, Honestly, I don't know because we haven't looked at the stats of how many people are using it. So it's hard to answer if it's useful or not, right? We heard from people that they really enjoyed it, but that's anecdotal evidence, right? We don't have like hard evidence to say, oh no, it helped four papers or three papers to improve their work. Our hope by building that is to make ease of access easier, right? So that if people can just simply go and if they're interested in a particular gene, they go in a search bar and say, here's my gene, and then here are the results. Is it good or is it bad with the gene I'm working on? So what we wanted to do is make it simple. So at least for that goal that we wanted, I think we achieved because people can just go search for the gene of interest, if it's on their patient or if it's in gene flies that they're using, or if it's a mice that they're working on, they can simply go search and see our results in comparison to theirs.
And you chose to submit the preprint to eLife. It's gone through the revisions. eLife currently experiencing a whole bunch of things, some good, some bad. Firstly, whose decision was that, and how did you feel about submitting to eLife?
Well, I'm not very familiar with the process, so I think Rion is gonna be a way better answer. But for me, I'll take anything. At the PhD level, it's a paper, it's published, it's nice.
Hmm.
So I guess the best person to have here to answer that question will be Paulo, right? Because he's the one that really spearheaded the communication and like the draftings of the details to make sure that phrases were correct for the journals we were sending. We have to be honest, it wasn't the first pick. We actually went into a very long and strenuous process with some really high stakes journals, and we had horrible experiences. We're not here to talk about which one they were, but it was a very painful process, right? I was the postdoc at the lab, and that delay in years in the publication, once we had everything ready, was damaging to a career, right? And I can only imagine that this happens more often than not in all the PhDs' careers, and not really great, to be honest, right? But then with eLife, I think at least the feedback we received from Paulo was actually really good. We got really good constructed feedback. The reviews we got were really good. They liked the paper. Had great comments about it. I think they really understood at a deeper level what we're trying to do. They really connected with the paper. And honestly, I think it was a great match. And maybe we should have gone there first. We wouldn't have waited for so long. And the paper would be out and people would be publishing or using the published data earlier in the process.
Yeah, I mean, that delay is a big issue, especially in the life sciences, but I imagine more broadly than that as well. And it is, right? It does, if you're an early career researcher, it can have huge impacts on your career. I'm all about naming and shaming, but that's probably why I get editors out of my own podcast as much as I do. So how did you feel about the language that eLife uses? So eLife, when I don't even know what they call it. I will call it publishing. When they publish their reviews for the preprint, they've got this sort of standard vocabulary they use, and yours was rated as fundamental, and the data was convincing. I don't know where that ranks in their wording. How did you feel about that as an experience? Because that's not something you would normally experience publishing. It's normally just a yes or a no.
No, I think I'm not sure about how postdocs feel, but for me, as a PhD student, it was very important to have more than a yes or a no. So to have people look into my data and stating what was very well done and reinforcing that, what was kind of okayish that could be better, and what they didn't want, they didn't feel like it made sense. And then constructing the answer to those comments, to that feedback, it was also a very important process intellectually, because I was not used to do it, right? You at school, at master's degree or whatever degree, you have a paper that you have to analyze, and then you have to simplify it, summarize it, and then you have to present it to a class. But putting at stake the work that we do and just stating, this is okay, so what can I do to make it better? And then having outside experts looking into it, because sometimes I feel a lot that we're very closed in our bubbles, right? And we only see the fine details of everything. And then we have to take a few step backs and look at the overall. We call it either the leaf or the forest. So it's very good to be able to look at the forest from someone else's perspective. So for me, it was very important, that kind of language.
I still remember when Paulo wrote us with what they actually wrote, and like he really focused, like they found our work fundamental, right? So which is I don't understand because this is not my field of publication domain from my past history of scientific papers, but that was a big deal, right? That they classified the wording used to describe our paper was fundamental. And I think that was a big deal that if you look at other research that's being published there, it was not there. And that was like a huge like stamp of, all right, our work is fundamental.
Oh, they'll really like that. I have a meeting with Damien who heads up eLife. I should tell him that. I think that's kind of what they're trying to go for, right? They're trying to get away from you published in eLife and more of that you published fundamental work. They'll like that. So we'll talk a little bit about careers, actually, I guess. How was the transition from academia to industry? That's something I know a lot of people currently trying to do. So how did you find that, and what advice would you give to people doing it?
If I could summarize it, I should have transitioned earlier. But the whole process, I think, was a really good learning experience for me. It all started when at the institute we were, they had a career mentorship type of program, and we had a session, and we went there, and there was one particular exercise we did that really resonated with me, which was the value matching. So it's a deck of cards. It's very simple, but it's a deck of cards that describes values, right? All the way from like your family values, you like to work in team, you like to work alone, you are excited about mental work, or you like physical work. Those values, right? Things, but also things that I would never think about it, that is a value that resonates with me or not. So it's good to have the prompting. And I started matching my own internal values with the values of academia at the stage of the postdoc trying to get a professorship position, right? And then as I start doing the matching, I thought there was very little overlap between my own values and the values that I find in academia at a professorship level. That's when I decided this is not for me. I have to match these values with somewhere else, right? And once that was clear, it was very easy for me to make the transition and go and search for positions that would be aligned with my values. And I must say, not all industries are aligned with my values, right? So, and I was happy to have found a company that actually my values match with the values of the company. That's not the case with every field out there, not every domain out there. The same with academia. Some domains in academia are more aligned with your values, others are not. Unfortunately, in my case, my field is not very aligned with my own values. I like to do teamwork. That's why I enjoyed so much work with Joana and Paulo and all the others. Collaborative, interdisciplinary work, it's at the core of who I am, right? I like that, and I chase it. That's why I went to the lab to talk to Joana and look at the microscope. I was interested, deeply interested in what they were doing. Um, and looking then at the professorship positions, like I would be alone. I would have to pull the rug from under other people to climb the ladder of full professor. And I don't want to do that.
Yeah, where I did my PhD was very collaborative.
And how difficult was the transition from academia to industry? Like, how hard was it to get a job, actually? Like, very straightforward.
Great question. So I think it's easier for me in the current job market, right? Because I come from technology, I come with strong coding background. I'm a good data scientist, right? And that is a easily translatable skill into industry. But not only, we in the PhD and also in the postdoc, we learn a lot of skills that we don't put a name to it, but are highly desirable in the industry section, right? We are independent. We can do research. We don't need anyone telling us what to do. We just take the problem, we solve it, and we present solutions. We are team leaders. We have excellent team time management of our own and of our teams. We are excellent bookkeepers. We are excellent writers. So that's a long list of skills that the companies need and the companies really desire, right? So it was actually much easier once I understood that I had a lot of skills that could be easily translated into what a company needs.
I think that's the bit people struggle with.
Yeah.
Yeah, so I was at a careers thing, and I was basically doing said exactly that, and that kind of turned people's heads a little bit because I don't think they'd quite matched up what they're doing with what the actual name for what they're doing is. Like doing a PhD, you're project managing, and that's really useful outside of PhD, but people don't seem to think of it in that way. Speaking of PhD, how's finishing up a PhD going? How miserable are you on the current end of PhD scale?
From 0 to 10. So this is a hard question. So I've learned very early on my academia path that I have ups and downs. So there's times that I'm super excited and I want to do it all at the same time, and I'm able to come to work at 8 a.m. and then just leaving at 9. And then I understood that was not a thing that I could keep going on for a long time. And then I have my downs where I ask myself, what am I doing here? Am I this good? Because I'm surrounded by very smart people. And then I think, so it's a bit of imposter syndrome that I'm just here because I'm lucky. So yeah, we all suffer from that. But I really like what I'm doing. So overall, if it was well paid, I would stay here forever. The financial issue is a big one because Lisbon is very expensive, and I've made the math, and I spend 73% of my income on rent. So everything else, it's very, very tight. And that's a big thing. People say, oh, don't do it about money. You have to work on what you like. Yeah, of course, you have to work on what you like, but you also need to survive to be able to work on what you like. So I do love being able to do a PhD. I'm very lucky because my parents kind of brought me here. They incentivized all my studies. I love doing it. I'm a big fan of bench work. I hate being on a computer analyzing data. That's not my thing at all. So I think I would be able to be a very good technician that has to be hands-on and do not disconnect from the intellectual part. But yeah, I'm doing it's being very hard at the moment. I still think I'm in a up at the moment. So that's good. I have a lot of stuff going on. I want to do it all at the same time, but then I have to try to balance it with, well, my social life and being able to be with parents, and every little change in my, well, family or friends or not professional setting, it's a big disturbance to the PhD because we have to do everything yesterday.
That's good for an end of PhD, I think. I think it's always a really stressful bit when you're trying to get a thesis, get papers, still do lab work, look for jobs.
So if I maybe make a counterpoint to Joana, I I work less now than I did when I did the PhD and the postdoc. I have a much higher quality of life because I'm able to focus more on the hours I'm working. Doesn't mean that sometimes I work until later or sometimes I have to start early because it's a global company. Sometimes I have calls with Australia and that's like 6 a.m. for me, right? So there's that, but it's not always that. And during the postdoc, the PhD, because you have your own self-drive to make sure you can complete a lot of work, you end up eating your weekends, eating your nights, waking up early, and just like overworking everything, right? So now I'm able to keep a much better balance between, all right, I worked some more this week, but the next week I'll get out earlier, right? I manage my own time so that I'm able to refill my mental stability if I work too hard on 1, 2 weeks, and then I'm able to cycle more. I like to cycle, so I go cycle more the next week, right? So that balance improved a lot my work as well, right? Because you work better, you're more refreshed, you can think better on the problems you want to solve.
Yeah, so it's good to know it will go away and I'll have a life again. It's gonna be good.
Yeah, it does. How do you find that transition in terms of identity? So one of the big problems I've seen, and I had this as well when I was leaving academia, is that I think a lot of people go into academia, especially maybe this is just a wet lab thing, but we go into it wanting to run a lab, wanting to, that is our life. We become so engrossed in it that it becomes who you are, and it very much becomes your identity. And then when you're trying to leave academia, you're not just struggling with trying to find a new job or a career path, but you're in many ways letting part of yourself go. How was that? Did you experience that? And if so, how was that?
That's a great point and one that I've thought about a lot about it, right? Because in academia, you think being a scientist in academia, it's all you can do, right? But I very early on understood that I'm a scientist, period. Doesn't matter where I am. I can be a scientist in academia. I can be a scientist, and I am a scientist at home. I am a scientist throughout my job, throughout the day. I am a scientist on the weekends. I'm always a scientist. Once a scientist, always a scientist, and that's who we are at a deeper level, right? So where you work doesn't matter. What I like to do is to teach, to research, to write my science, to talk about my science. And this I can do in academia, I can do in the company, and I do at the company or anywhere else. I do when I talk to friends, because we're all, of course, scientists have their group of scientist friends. So that's what we do when we talk to each other, right? I don't go as much to scientific conference anymore, but we have internal conferences, and that is I learned to accept that there's the trade-offs, right? It's also important that when you search for a job, you need to find the job that will let you be a scientist, right? Sometimes it's not the first one. Sometimes it might be you have to grow into it. But once you put into that mentality that that's who you are a scientist, period, you will grow into a position that will let you be a scientist naturally. Even though if you haven't started at one, you can start as an analyst, and then people will see you as a scientist and they will move you into a more science-driven position, right? But you can't forget that. That's who you are. You are a scientist, period.
Yeah, that's good advice for anyone listening. That's where we should end it. All right. Well, thank you both.
Thank you.
Thank you, guys. This was lovely.
And that's the show, folks. If you enjoyed listening, then hit that subscribe button to get the latest updates straight to wherever it is you're listening. Don't forget to rate us on Spotify. Until next time, have a good week.
This podcast was produced by Rippling Ideas, turning ideas into waves.